to the Weekly Bioanalysis, a KCAS podcast.
Hello and welcome to the 61st episode of the Weekly Bioanalysis, the official podcast of KCAS Bioanalytical and Biomarker services. KCAS is a bioanalytical CRO servicing the pharmaceutical and biopharmaceutical industries for over 40 years. My name is Dominic Guarino, and I've been in the ligand binding assay and cell and gene therapy space for over 28 years. I'm here with my co-host, John Perkins.
Hi, I'm John Perkins, and I've been in the bioanalytical LCMS space for over 28 years.
And if you hear a knock on John's door, that's because it's Halloween and it's getting late here. No, John and I are senior scientific advisors.
If you hear a regular drum beat, yes, I've got visitors.
John and I are senior scientific advisors at KCAS, and either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. I'm located in Kansas, John is in upstate New York, and our producer is in Missouri, so we're all over the map. We are amped up to have you here on Halloween to listen to the 61st episode of our podcast, which is now available virtually everywhere. Wherever you choose to find and play your podcast, you can now likely find the Weekly Bioanalysis. We welcome all of you, whether you are joining for the first time or if you're a regular listener, to give us some feedback. As always, today's podcast will be a review of our latest news and resources, and then a focus on a topic of our choosing. Before discussing any feedback we've had from you. We're constantly looking for topics, and we'd be happy to discuss something you want us to cover. So again, we're amped up to have you here, and we are looking forward to a fun episode. To kick us off on podcast number 61, John is going to go over today's podcast topic a bit before we jump into the news and resources section. John?
So, as usual, we'll do the COVID and non-COVID news items and then move into our main topic, which is a discussion of the 2022 conference season and KCAS's participation. Let's move on to the news.
So, John, why don't you tell us the latest about our news and resources? As always, we'll start with some COVID. John, what's going on the COVID front?
So let's start on what could be potentially helpful. FDA hands down emergency okay to plant-based face mask, and this is interesting because you don't necessarily think of the side effects of widespread face mask use, etc. But of course, with people using them just as a disposable basis, we generated a lot of face masks that need to be disposed of, and there's a study in 2021 estimated that 3.4 billion single-use face masks and shields were being discarded daily around the world. And in a reaction to that, the FDA has given emergency use authorization to a single-use mask from Canada-based PADM Medical, where their masks are actually plant-based and as a result of that compostable. Although you can't just stick them on a compost heap in your backyard. Composting comes from taking the strings off, and then it's submitted to an industrial composter who can then break it down in a commercial compost center. But it's still a step forward. It's still a good thing in comparison to, I don't know about you, but when I walk around here, I regularly see just, face masks lying in the gutter and things like that. And anything that can counter that is a good thing in my book.
Yeah, I actually saw this one when it kind of came out and thought, wow, this is very, this is a case of, we created something, let's fix it, right? So there's some staggering numbers. I mean, the 3.4 billion face masks or shield discarded daily. So we definitely need something, John. I think it's great. Hats off to PADM Medical in Canada. I couldn't really tell from this how effective they are. They, I don't know what they're not. Are they as I think the natural question is are they as protective as, say, a surgical mask, which would be suitable? They're not
I,
the M90 type
No,
protection.
they're not the N95. I think they're like the more protective ones that if you go to a hospital they'll give you.
Yeah,
So it's more than a cloth mask, but less than the N95.
yeah. It's good though. I mean, these are like, John, we do, to your point, I do see masks periodically. I mean, even today, there's still people in the building wearing masks. I think you'll see people wearing masks and going forward. And so these types of environmentally sound masks just make sense. And whether they're like, able to withstand gases from any sort of biohazardous gases and so forth, no. But can they help with knocking down your probability of making others infected with COVID or you getting COVID? Absolutely. So
Yeah.
hats off if you want to wear it. The question becomes, can we get them to a state where they're maybe edible afterwards? No, that was a joke.
You want flavored face
Yeah,
mask?
I
Yeah,
want
I'm
it.
not sure I
Remember
want to go there.
like a fruit roll up type version of it. No, I'm just kidding. No,
But
John, that's a really good way to kick us off here because even we John, we talked about maybe even altering, like, and we got more COVID stories to say. It seems like, is it really still in the news? But if you have anybody who's a college student right now, everybody's bracing for COVID's going to start breaking out. And it's not like, remember, every college has an influenza outbreak, has an
yeah,
outbreak here and
yeah,
there. And so I don't want to be an alarmist, but like, that's where we're at now. We're returning to normalcy. There's no more virtual classes. You got a bunch of adolescents or teenagers that are now wanting to live like we did when we were in our first couple years of college, which means in a dorm room or in a dormitory, never the cleanest places. And it just seems like we're probably in for some more outbreaks on college campuses with COVID, and that just means we should be thinking more and more about getting plant-based masks to them. So, John, I'll let
sure.
you go on to
Yeah,
the next one
and
unless you had
it's one of
something
those.
else. But yeah, go ahead.
It is one of those things that, yeah, as we go into winter, that's when people go more indoors, and if it's poor ventilation, then there may be transmissions. Seems relatively quiet around here at the moment, so
Well, good. I hope
yeah,
it's the one. Yeah, sorry.
I
Go ahead.
thought I thought the footer for this article was interesting because it sort of says this is actually part of the FDA's focus is looking for environmental sustainability, and so they're also looking at things like biodegradable medical gloves, with the example being one that was developed by the Showa Group, and these gloves are powder free, made out of nitri- nitrile butadiene latex, which doesn't trigger allergies to national rubber latex. In lab tests, the gloves took just over a year to break down by at least 80% in an active landfill, and expecting to reach full decomposition between 1 and 5 years, much more quickly than decades it takes regular nitrile gloves to break down. So that's something for us to look forward to as a look at as a obviously a company where our glove usage is high because of the nature of the business that we're in.
Yeah,
I like the fact that there's the FDA is really looking at this as a path forward.
that's great. 510(k) clearance on those gloves, and then the face coverings are still in the emergency use. But you would imagine over time, I mean, that's just a staggering amount of masks.
Sure.
It almost makes me think like how resourceful we are that we can make so many masks. But
Yes. Yes. Indeed.
we'll move on to our next one, John. Right? Well, I'll kick us off here, and you can tell us more. It's major worldwide study clusters long COVID symptoms into three groups, and this is interesting, John. Long COVID is definitely something we'll be dealing with. But why don't you tell us a little bit about what I think it's the World Health Organization put this out, right?
Yeah. This is actually, yeah. It was the well no the it's basically a study by GMA Network study corresponding author Theo Voss, and he talks quite a bit during this article. And it's they basically analyze the data, and they're really saying that it sort of goes into three clusters: persistent fatigue with bodily pain or mood swings, cognitive problems, and ongoing respiratory problems that persist at least 3 months after acute infection. That 3 month cutoff comes from the WHO, so that's where the World Health Organization comes
Gotcha.
in. I think it's just in. So they basically said that 6.2% of 1.2 million patients from 22 countries who exhibited COVID-19 symptoms in 2020 and 2021 experienced long COVID. As you dig more into it, it's really interesting that it's actually women who seem to suffer more from long COVID than men, and it sort of correlates to the higher instance of like autoimmune diseases amongst women, like lupus and things like that, compared to men. So it's, I mean, all in all, it's just relatively interesting. I think the fact that long COVID has had attention, but not a lot of attention. I think it's going to become increasingly necessary to dig into what's going on. And this is really the first attempt to sort of try and break it into buckets and then diagnose it in terms of severity and impact on life compared to diseases as a whole, which is a way they sort of measure what these kind of conditions, what the impact are on people. So it's just an interesting analysis of long COVID, and then obviously talking about potential paths forward.
Yeah, and it's interesting. They, persistent fatigue, cognitive problems, and ongoing respiratory. And the persistent fatigue one, they lump that in with bodily pain and mood swings, in part because they very cleverly use the dengue and Ebola health statuses, right? So they're called ECOG statuses in the cancer patient world. East Coast Oncology Group gives a care of life 1 through 4, and they kind of did something here to kind of characterize, right, the how your life has been altered by this long COVID. And those 3 buckets, I think there's still a lot. What I'm driving at is I think there's still a lot to be learned here, John, because
I
I don't know
agree.
you, but I still encounter people that are like, there's a high percentage of people that are still got COVID, maybe never got it too bad, or was bedridden for a day, or even bedridden for a week, and then they've recovered. And then you've got those people that are like, yeah, I still can't smell, or I've still
Yeah.
got some motor skills, and it's really like, it's just if I don't know how I could take a side note here. I have a relative of ours, one of my wife's cousins, a close family member. She got West Nile's virus like 5 years ago, and she still has problems walking now. She's almost like she's disabled, basically, if that's the right term. I hope I'm using a
Sure.
politically correct term there, but she still has trouble walking, and for the longest time she couldn't talk. And some of it is, you have to rehab harder, and we've wondered about that. But it reminded me of like how finite your body is when it comes to getting some of these infections that, COVID falls into a similar thing as any sort of lingering disease. And the difference is West Nile, right? That affected 1 or 2, maybe 5% of the population when we had that outbreaks. I think it's almost 7 years ago now. But we have these outbreaks, but COVID was like mass quantities of the population. So one could expect more of these long COVIDs. And now that I know there's even companies trying to treat it and things of that nature, but I think long COVID is something that this gets back to no COVID parties. You don't want COVID, right, John? It gets back to that.
Exactly. Yeah. Yeah, exactly. Because you got no, if you get infected with it, you've really got no idea how it's going to affect you. And I think that's the, if you get infected, you come through at the other side and you feel okay. You thank your lucky stars that you're not one of those people of the 6.2% who've
Yeah.
gone on to really have much longer, effects of this disease. I mean,
That's what,
it's
that's
definitely not something I would ever advocate trying to catch.
Yeah, it's 1.2 million people, John, that they estimate. That's not
No,
that's way,
they surveyed 1.2 million people, and it was 6.2% of those
oh, got it.
1.2
Got it.
showed
So that
long
means,
COVID.
wow. Wow.
That
Sorry.
gives you an idea that the numbers could be. I mean, the numbers could be huge on this. If you talk 6.2% of people, if that extrapolates to the general population, that's a lot of people.
Well, I think it'd have to extrapolate to, people who got COVID, I don't know, and had one of these, you'd have to fall into did you get persistent fatigue,
Yeah,
cognitive
yeah, yeah,
problem, or
absolutely.
ongoing respiratory? But I mean, I just think there's, man, it is something that has got me on Halloween thinking about how, we think to some degree, a lot of people, maybe even myself included, have fallen into this comfort zone that COVID's just going to be the flu. Well, last time I checked, flu didn't have these long COVID as well as some of these persistent fatigue, cognitive problems, and ongoing respiratory. I don't know of anybody that has had anything like that. I'm sure they're out there, but for the most part, you just recover, and certainly not at 6.2%, because that's an astronomically high number of people if they just base it off of 1.2 million over 22 countries. I see it now. I mean, what are the staggering numbers of people? The number of infections with COVID is in the billions, I think, right? I might not be mistaken there. So that would be
one thing about this study is based on the Delta variant, whereas we're now in the variants of the Omicron variant. So the numbers may be very different for Omicron.
There you go. That's a saving grace too. Maybe it's less virulent maybe some herd immunity's kicked in a little bit, so maybe it's not quite as high with the future strains of it. That's logical. But one last point here is I find it interesting around the women are more susceptible, because if we remember, women were more protected against COVID, right? So that's a little bit interesting around the infectivity of COVID is lower in women, but when you get it, you're actually, and maybe that makes sense if I can talk my way through it, right? Because it was all due to hormones. At least I believed it was a hormonal thing that helped protect women, because it has to do with that ACE2 receptor. And I still think we don't research that quite enough. Anyhow, interesting.
So, just in response to that, the study authors said that X chromosome-linked genes are thought to influence susceptibility to viral infections as well as autoimmune diseases, lending support to autoimmune processes playing a role in the development of long COVID. So that was one of their conclusions, and that's why they're thinking the discrepancy towards women rather than men.
True.
So, shall we move on?
We can move on, but in my simple mind, I still think it's hormonal. No, I'm just kidding. No, sounds good, John. Yeah, what's up next?
So, next up, Pfizer BioNTech partner with Marvel for Avengers and COVID fighting comic book. So, basically, in the Everyday Heroes digital comic book, an evolving Ultron, one of Avengers' leading villains, is defeated by Captain America, Iron Man, and others. The plotline and history of Ultron is explained by a grandfather who's waiting with his family at a clinic for COVID-19 vaccinations. Although not explicitly spelled out, the evolution of Ultron and the creative solutions the Avengers keep coming up to block him mirrors the story of Pfizer and BioNTech's scientific efforts to deliver vaccines to fight COVID-19. And after the Avengers win the day, they fly by the window of the clinic and say hello to Grandpa, aka John, who turns out to be a retired damage control foreman for the team of superheroes. So I thought it was really. I always like how. I mean, obviously, there's a lot of people in these companies who are thinking marketing and all this sort of stuff, but I thought the link up of Pfizer with Marvel is just. I just thought it was just interesting to me. I'd love to see a copy just to see what the whole story is like.
Go ahead, John. Sorry.
So I mean, it says John then links the Avengers battle to the COVID one, saying, "And that's how I know that we can fight back against even tougher, ever-evolving enemies. If you're willing to adapt, fight back, and take steps to help protect yourself, it's also important for entire communities to come together and help fight the threat." And that's exactly what we're doing today, his middle-aged son says as a nurse calls the family back in for their shots. So, so I mean. Man's mind is very interesting. The different directions that everyone goes in, and I just thought it's. I mean, the script was written by Paul Allor, who also worked on G.I. Joe and Teenage Mutant Ninja Turtles franchises. And he posted a link to the book on Twitter, adding that he's getting his bivalent booster and flu shot tomorrow. So it's that was
that's great, right? Like, I mean, Marvel is
out for if you're interested in all the way COVID's evolved in terms of how we talk about it, you know, how we try and persuade people to go and get vaccinated, all the different tacks that are taken. I think this is a really novel and a little bit out there approach, but. You know, if it persuades a few more people to get vaccinated, then that's great.
Exactly. And it will. This will work.
Mm.
I mean, Marvel is absolutely the probably the best movie franchise. I'll say it. I love Marvel movies. They're wonderful. Captain America is one of my favorite. I even like Iron Man 2, which a lot of people don't. I enjoyed the Iron Man movies as well. And so, the Ultron, I think that's the Age of Ultron was a movie. That was the second Avengers movie, I think. But you nailed it, John. This will get people to get vaccinated just because, I'd like to see it in doctors' offices and wherever it might be, just to get people interested in Marvel sells. Just walk around the mall and look around and see how many Marvel shirts you'll see on it.
Exactly. Yeah.
It'll be
Yeah.
more than after the Kansas City Chiefs here. That'll be second. But and then there's what I didn't realize is this isn't even the first time it's been done, right?
Yeah. Yeah.
So,
Takeda did it before, for a series of books on inflammatory bowel disease.
one of my
So
favorite topics too, John. You know, I love a good IBD talk. I actually studied it for a semester, half a year in grad school because it's such it was a fascinating way to learn the immune system for a couple different reasons. One, the mucosal immune system slightly different, but it was such a it's I don't know, like 1 in 3 Americans have some form of it as you age, maybe even half the population, and then it's very treatable. I digress there, John. I had to
sure.
talk a
Yeah.
little bit about
Yeah.
bowel disease. No, and then Novartis did one too, right? They had
Yep, 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 and that was more of a homegrown effort. But I think any of these initiatives where it's like helping educate people by whatever path they take, I'm always in favour of it. And I know that we talked in the past about other other initiatives to teach. The Novartis one was about rare disease, and we've talked about other initiatives to teach people about rare disease and the impact on families and things like that. And I think they're all good.
Yeah, and John, just thinking about the future of COVID, it seems like we'll keep talking about it. I hope someday we don't have a COVID section of our news.
Oh, I agree. It's interesting that a few months ago it seemed like we didn't have much to talk about with COVID, and then the last couple of times it's been some really interesting news items about COVID that really sort of want to get me talking about it again. But I'd love it if that section went away because it's be indicative of the progress that we're making.
Yeah, I agree, and I think when I say go away, it's just add it to the list of, sort of battery of things you gotta kind of stay healthy for. Just it's
Yeah,
also,
yeah,
the idea of washing one's hands and social distancing as much as possible, or even like, even if it's not 6 feet, just don't let people sneeze on you, for instance, or just whatever we were doing pre-pandemic. We should never really go back to those types of behaviors. But in the same token, you can still shake someone's hand. It's just don't make sure you wash your hands if you're doing those things before you say stick it in your eye or I don't know, John, but let's move on to the non-COVID issues.
sure. So it's non-COVID time, and this headline is "Don't Junk DNA as Study Shows New Role in Brain Disease." So basically, junk DNA was coined as a phrase was coined in 1972 to describe 98% of the genome that doesn't code for proteins, but actually research is continuing to show that it's obviously involved in bodily processes, and the most recent research is from the University of Sheffield in the UK have discovered that non-coding DNA also breaks, and if it isn't repaired can contribute to the development of neurodegenerative disease. So they hope that this can help identification of biomarkers, but also give another path to possibly come up with new therapies. Because as we know, there's a lot of focus on neurodegenerative disease, but there's not a great deal that's really been successful in terms of treating it. So it might be that this gives a different approach where you've got a strategy to tackle things.
Yeah, it's just just makes us realize how much and we know a tremendous amount about the brain and neurobiology in general relative to where we were, say, 10 years ago, 5 years ago, even last year. But this 98% of the genome falls into this sort of what we thought of, at least I learned it as junk DNA. And then even in grad school, they said, well, it's not junk DNA anymore. It's that DNA, the way it can support it structurally for the other 2%, right? These are all exons, introns. That's what we're talking about here. So there was believed
Yeah.
to be some like structural importance about it. And now even reading this, I'm like, man, it makes sense that you can't just nick this and think it's not going to impact or have damage to it. And it's not going to be a player. And you talked about the really what probably is one of the best fallouts that this not fallouts what they'll probably start to reverse engineer is how I think of it is the biomarker aspect of it, because there was all these A beta peptide biomarkers. Synuclein comes to mind, BDNF, NFL. These are all biomarkers that have been in any sort of neurodegenerative, I'll say Alzheimer's and Parkinson's and things like that. This could be the real way is if you could see conceptually, for me, I could think of, okay, if you could take even just someone's cells and do a characterization of this so-called junk DNA, then that could potentially lead to just an overall understanding of the of your health and so forth, and then even like some soluble ones that this pneuma, right? That's the protein that might fall
Yeah,
out.
so yeah, I'll talk through this actually. So the researchers showed that non-coding DNA acting as promoters were prone to strand breaks due to oxidative stress from cell metabolism. The NUMA, which is nuclear mitotic apparatus protein, was actually is mediates repair of the strands. But if you've got low levels of the pneuma, then oxidative damage and strand breaks accumulate, leading to dysfunction and ultimately opening the door to Alzheimer's, dementia, MIND, and other conditions. So that's what they're seeing as the process.
Yeah, it's really, I think there's still some to be determined around it, but this is what the field needed, a new approach and some
Sure.
new
Yep. Yep.
a breath. This is a great story, John. This is the one that in a few more years, these are the ones that we want to follow up and see what the progression is. But definitely a good way to kick off our non-COVID news, John. The idea that we could come up with a better way to tackle brain disorders.
Yep. Absolutely. Yes.
Well, all right, we
So
got a couple more here. Yeah, go ahead. Tell us about some pain
let's
relief.
move
Yeah.
on. I noticed something popped up on my screen, that was all, and it slowed me up. This one is pain relief without opioids ill effects, new molecules found. And this is actually small molecule stuff, so it's which is nice. But obviously, we're all to a large extent, we all take pain relief for various different conditions. But when you get chronic pain, there's it's obviously it's much moved people much more to opioids, and then there's the long term potential for addiction and just deleterious effect on your life with those. So they basically these are UCSF scientists funded by a DARPA grant have identified a new set of non-opioid molecules that ease pain without sedation. So it's they are agonists of the alpha 2A receptor, which, when activated, has pain relieving effects. It's the same one targeted by heavy duty sedatives, dexmedetomidine and clonidine, which are commonly given to hospital patients who must undergo operations or mechanical ventilation. But unlike those drugs, new found molecules don't have sedative effects.
Hmm.
So it's basically Brian Scheuchert,
Sure.
who's the study co-author, said that breaking apart the pain relief and the sedation means that you actually have something that could be a promising target for drug development. So again, it's early stuff, but it's just, I think part of the reason for really wanting to go in this direction is because of the effect of opioids, and obviously we have a ongoing concern about opioids on the street, etc. But it means that people who actually need those pain relieving drugs often have their access to them limited because of prescribers are being really, really careful about prescribing medications. So if you've got something that doesn't have those addictive effects, then the potential for it in the long term is huge.
Yeah, this is, that makes me think of the cannabinoids. There are now
Yep, sure.
treatment of using cannabinoids, but they don't get you high, right? This is a similar sort of. It's almost like a holy grail. I don't think they're quite there yet, right? Because you mentioned some
No,
heavy,
no, no, no.
yeah, clonidine, and those. I mean, that is like big time stuff. But this is like some return to normalcy, John. What I mean by that is we've got companies now, and even we're learning about things that are small molecules, or in this case, it's sure pain meds, but it's not COVID, and we're back into like treating other disorders again. So that's the one plus side that popped into my head. And then obviously the altruism of this is, what better way to avoid the opioid endemic or pandemic or epidemic or however we think of it than to have something that just simply has at least some similar benefits. Again, I think we're a little ways away from being able to take something that's going to give you that much pain relief and not get you sedated. But it's a step in the right direction, and this is
Yeah,
where we like
so
to see
so
the
this,
research
I
dollars.
mean,
Yeah.
this is at the mouse stage, so it's got a long way to go. But it's basically this is what they did. They looked for basically drugs that were going to interact with the alpha 2A receptor. They actually screened 300 million molecules to look at them in terms of size, and were not chemically related to existing drugs. Then of the thousands that came up, they used computational docking to see which were literally the best fit. Narrowed it down to 48 candidates, and they then tested those in human and mouse cells and found that 6 were likely to activate the receptor. So then these were moved to testing in mouse models for chronic and acute pain. In all of them, the drugs relieve pain without the sedative effects seen in other drugs in the same class of molecules, even at much higher doses. Because they assessed that because there was no reward seeking behavior by the mice, in terms of they weren't doing anything to try and get more administered to them. So
Yes. Yeah.
that's what it is. It's early stages, but it's sort of it points to potential hope for the long term. So I thought it was really interesting.
Yeah, I want to rehash some of those numbers. Are just amazing, right? 300 million molecules is what they had to scan. And from that, like you said, they broke it down to 48. From the 48, they found 6, right? So 48, they actively start to test in vitro models. Then they break it down to 6, and it looks like they got like 2
They got two.
molecules in particular. Yeah. And
They
it's
got
just, go ahead.
they got 2 they really like. So
Yeah.
300 million to 2. Yes, you're right. It's
It's
whoa.
staggering, and then like it's lifted me up a little bit. It's making me think we're starting to get back towards addressing. You hear about the fentanyl issues in the U.S. and all the reasons for it and some of the solutions for it, but really it starts here. Just we wouldn't need those things if we had something that didn't give you a high. Yeah.
Yes,
Right?
yes,
There
yes, yes.
wouldn't be if we had something that could replace it. So this is far upstream than what I think the average or every politician or talking heads talking about. You can forget all that. If we just make a better pill, we're
Yep.
good. Yeah.
Hmm.
Okay. One last
Exactly.
one, right?
So
Let's wrap it up. One more though. Good one here.
this final one, yeah, potential potential for broad spectrum antivirals could lie within host cells. So this is basically talking about antivirals where things like hepatitis C and conditions like that. It's really you're developing a drug that basically addresses one condition and that's it. But a team of researchers from Ohio State University have identified an enzyme that, when inhibited, slowed viral replication and improved outcomes in mice infected with a wide range of viruses. So it's actually there's a possibility that this targeted enzyme NSUN2 is which is involved in 5-methylcytosine methylation, which is in multiple biological pathways, could actually lead to it could actually help them identify something that could cover a number of different viruses to treat with a single drug.
Yeah, I think this is part of, look, I mean, I can break it down on cervical cancer, right? Cervical
Hmm.
cancer is caused by the human papillomavirus. And theoretically, you can have one form of the papillomavirus, 16 or 18 of the two major ones that go on to cause cervical cancer. But um, the number of women who have one HPV strain to them that get cervical cancer is very low. It's typically over, I think, I forget the numbers in there, somewhat dated, but it was like 75% had either two strains. So that's kind of I think that's what this might be driving at. It's like, why are we going after someone with COVID with just a single COVID vaccine instead of trying to treat them systemically? And it's a big thing, right? Like, it's not a simple, we want to simplify it, one bug, one drug. It even says that in here, right? But that concept is
Yeah,
probably part of why when someone says, how come you don't have a cure for cancer? That's kind of it, because I jumped into the cancer realm of things, but why don't we have more cures for infectious disease? It's probably because we're treating somebody with, say, COVID, and really, not only do they have COVID, they've got these, and we know this is true. If they have underlying health conditions, that impacts it, right? Are you overweight? Are you a smoker? Are you blood pressure? And that's for scratching the surface. Those are the easy ones to look at. Never mind genetic disorders or even other sort of like, there's no test that says, okay, you've got COVID and these 6 other viruses. Oh, if you've got COVID with Hep C and this and maybe a respiratory virus, oh, you need this, right? That is that making sense? What I'm saying, John? Like, there's no like that. That's a wishful day of artificial intelligence may get us there, but first we need a test that can take
sure.
just a tube of plasma from somebody and give you can we test 1,000 different viruses? And then it reads out, okay, and just the other part is latency of viruses, which is even like a really big thing to talk about, because we're all really CMV infected or even Epstein-Barr infected, or at least had it. That's part of our natural immunity, right? So like, if there were a test that said, okay, well, John, you've got these 6 viruses that you're protected against, we want to treat you this way, but Dom, you've only got 3, we want to treat you that way. Man, that's like Star Trek stuff, right? That's where I think our field is heading, and I don't know if this gets us that way, but it sounds like it is. That's where we have to.
Oh no, I mean it is definitely out there, which is why I wanted to highlight it. So basically, this enzyme NSUN2, it's like if it's blocked, then you stimulate the immune response. So, but having suppressed levels exposes host RNA snippets, and then your immune system sees them as foreign invaders, unleashing a type 1 interferon response that when it's at a certain level tackles the virus at the same time. So they're actually the next step for this group is to look for drugs that actually controls the suppression of NSUN2 functions, so that then they can see if they can use that to tackle multiple viruses. So that's the way they're coming at this. So it's looking at something, using something in the body to really trigger a response against multiple invaders.
Yeah, it even drills into some of the disease states they're going into, but this is also the paradigm of how. So I always think, my background's more in the oncology, but these infectious disease things, they face the same, as does transplantation, they face the same immunological challenges. And that is exactly what's happening here. The bug is starting to win a little bit. The innate immune response is not robust enough, and the virus persists, and the patient is sickly. Same thing happens you've heard the term acute to chronic inflammation. This is the same problem with inflammation and what causes eventually, certain immuno-oncology disorders as well as autoimmune diseases. This is the same concept, John. It meaning basically what these bugs are doing is causing the immune system to go from acute production of good interferons diminishing those, and by default it goes into the not bad ones. They're more regulatory or suppressor because they think it's remedied, that it's taking advantage of the own T cells. So I mean, I know I'm taking a leap on here, but that's why we're talking about it, John, right? That's what the whole thing is. This goes in. This is wonderful stuff. And I think the world is, the whole field is getting more and more in sync, and I hope that I've made a good case that acute to chronic, persistent infection to succumbing to the infection, right, versus remedying it, very similar.
Yeah, yeah. So I always like with the non-COVID to try and see where we're going in the future, because there's so much research out there that's pointing where it may take years on some of these, but you've got these early flags that say we've identified a potential future for where this medical field.
The good thing is this can move a little quicker because they're going into like cattle, right? That was one of the, so they have an opportunity, and that can accelerate medicine quite a bit. And I'm not saying they're going to do bad things to cattle, but they know exactly what they're dealing with, so it's a more controlled experiment than, say, a true, the human population, which is much more diverse than the cattle population.
Sure. Yep.
More controllable. Exactly. Cattle are in a certain state. You and I, just by default, you being in upstate New York and me here in Kansas, we're going to have different environments. And I know cattle do, but in general, cattle live in a certain way and eat a certain amount of food, right? They're whether you're a rancher in North Dakota or a rancher in Texas, you're treating your cattle very similar.
Sure. Yeah.
Yes. I wonder. Okay, I digress. What do I know about cattle? We're going to move on, John, now into our main portion, right? What an exciting topic exactly. We have, right? It's conference season, right, John? Our favorite time of the year. Where do we want to start, right? For our audience, if you didn't know, this is the late September through October time of the year is when a lot of major conferences are. It is a jam-packed time of year. I think in part because it falls into that sweet spot in the year where everybody's kind of back at school. We haven't hit any major holidays. Obviously the weather's great, so you can travel to any city. So, John, we got a long list, and do we just want to jump right in, or do you have anything else? Because I'll set
I think you're right. It's like you got universities back in session. It's coming off a summer holiday where everyone's back on what is a more of a routine. So it's good timing for conferences to try and attract people. From our perspective, it's another mechanism for us to be looking for business and also talking to people to find out where we can possibly help in the long term and actually drive where our business goes and find out where the industry's going. Where if the industry's going in a certain direction, how can we then contribute to that? It's we cover, well, obviously as we've added more services, we're starting to cover a more diverse range of conferences. You've got some in the calendar like Pharmacy 360, which is a much more general conference and covers tons of different bases. We have others that are much more focused in terms of like WRIB and EBF, which we'll touch on both of those. Really focus on the regulated aspects of bioanalysis. But then with the acquisition of Flowmetric and our own in-house flow cytometry efforts, we're getting much more into the cell and gene therapy conferences and having a presence there. And obviously part of it is getting our name out there. It's getting your name out there and it's talking to your peers and figuring out where we can help drug development. So it's a really valuable season for us.
Yeah, it is. It is not only, look, we're a business and it is good for contacts and leads, but we have two major meetings. You touched on them, WRIB and Pharma 360. Those are really, WRIB is, if you're not familiar, we'll just jump in, right? That's the workshops and regulatory issues in bioanalysis. And that is the number one meeting, in my opinion, in the continental US for our quality and regulatory aspects of bioanalysis. So it's the who's who of regulated bioanalysis. I've been going to this meeting for 9 years now. As an organization, we have been to every one of them. I think this year was
Yeah.
the 17th or 18th. I forget where we're at. Or maybe it's not that many. Maybe it's 15 or 16, but we've been to every one of them. So that meeting, I mean, we put a lot of effort into it beforehand. We send a large contingent of people there. It has the global CRO council ahead of it, right? That's another piece. That's the GCC, it's called, right? That's another
Hmm.
part of that meeting. And not only is it a great area for us to make business contacts, but really that is the premier meeting to go out and what are we seeing regulatory wise? What are the issues we're having? It's a great collaborative meeting to talk about it. There's so John, I'll take a breath there, and I know maybe I don't know if you want to touch on the EBF at this stage, or do we
Oh,
want
no,
to?
we'll hold that for
Okay.
later
So
because I'm gonna give you my WRIB experience is zero. this was this was going to be my year to finally attend it,
well,
and
I
then other issues intervened, so I had to back off, which
well,
is
that's funny
I'd
that
love
you'd never been, John. I guess it is. It's still heavy. It's
I,
not small molecule, large molecule, but it is definitely got a bigger large molecule theme to it, in part because of
yeah.
this immunogenicity aspect, right? So we'll take a small step back, make sure the audience understands. We know there's small molecules and large molecules, or what we call pharmaceuticals and biopharmaceuticals, and then we have the in between in our hybrid, and then cell and gene therapy, which has quickly crowded this space, but they've spawned out their own meetings. But within the large and small, you've got pharmacokinetics, which is really the backbone, right?
Mhm.
Regulated
Yep.
PK analysis is the backbone of drug development and bioanalysis. It's still probably the number one, not probably, it is the number one thing we do at KCAS. And right. So that's still across our service lines. That's got, I don't know what it is, but I bet you it's still a big chunk of our work. Maybe something even to look up, John,
Mhm.
right? But then what is we've harped on this before. The biopharmaceutical, the large molecule has this second aspect, which is as critically important. That's the immunogenicity piece. So by default,
Yeah.
now that there's a rival method, and we're not going to talk about biomarkers because they're not really discussed at WRIB, certainly not as much, but those major regulatory things, by default, it becomes more of a focus point now because WRIB has to incorporate immunogenicity as part of it. So there's this like contingent of small molecule people who don't want to talk about that at all. And then there's this large molecule people who are there solely for that. But it's become a big forefront, and why I feel like maybe that's answering why I've been to so many more of them, because the immunogenicity landscape is changing annually every 6 months, where the PK, it's changing, but not. And I'll give you an example. We now know that in a preclinical setting, if your drug is a monoclonal antibody, a low risk immunogenicity, you don't need to the FDA is now saying you don't have to fully validate those things. Well, that's a major change, John. And so that's something
Hmm.
that is
Yeah.
definitely, it was discussed at WRIB this year for sure. So maybe I'm beating WRIB and we can move on to the next one, but that was an important
I,
part of
I,
it. Yeah,
I'll
any
give a
final
little bit.
comments? Go ahead.
I'll give it a little bit of perspective. The reason why I haven't been is obviously I was in another company before KCS, and we had a different. It was there was a contingent went every year, but I was never selected to go. But that was largely because I went to EBF instead. So in terms of which we'll touch on later, but
Ah,
if you talk about the number of times you attended WRIB, I've been to at EBF since the beginning, and it was also affected by the fact that I for a few years I was living and working in Europe because we had a lab in Europe that I headed up. So that's all played into it. And then by the time when I joined KCS, when I probably would have gone to WRIB, we went into COVID, so the opportunity went away. So that is one thing that's
yeah,
worth saying. This is the first year it's been in person since COVID happened. If I remember correctly,
it is.
they did virtual conferences, but this was the first in person one. So
First time in 3 years, correct. Yep.
yeah, yeah. So I mean, yeah, it's like you say, it's very much the bioanalytical conference to go to, in the US, and people come from all over the world to it because of its importance. It's just so happened that I haven't.
Yeah, and then John, do you have we can move off of WRB. You got like the next conferences you want to talk about, or what do you want
I
me to?
mean, we can talk about whatever you want to talk, whatever you want to go through. I mean, we some to some extent we're well into conference season right now, so a lot of these were looking back on what's done. Don Duffield went to the Gordon Conference on lipids, led panel and things like that. The Gordon Conference or a series of conferences on different topics, but we don't have much. We haven't really interacted much with those. I think that's something, if you've got a specialist field, we may have more participation in that, but it
One
really
of the
depends on the topic at a
Yeah,
time.
they're one of the longest running conferences. Gordon conferences are very prestigious, and that's part of why
Mm-hmm.
Don went. It is usually a
Sure. Yeah.
slightly more academic sense to it, but it's more cutting edge and usually pioneering type things, and that's for sure what Don was doing at this one. Just going down our list, maybe before we jump into immunogenicity, let's just wrap up Farm 360, which is formerly was known as AAPS. That's the American Association of Pharmaceutical Scientists. Now it's referred to as Farm 360. This was at one point in its heyday. I think there were close to like anywhere from like 12 to 13,000 people at it. I
It was,
heard this.
it was huge. Yeah.
Yeah,
Yeah.
it was a large conference. I remember I've been to maybe 7 of these or 8 of these, and I remember one year, very large one in Atlanta. We rented out the entire aquarium down there. It was quite a shindig. There was easily, I think, close to 10,000 people at the reception for it. This year's conference, I heard some preliminary numbers of about 6,000, which is still nice. That's a big number. I heard it was very good. I didn't get a chance to go. I had a conflict, but those are meetings where, a good portion of it is true pharmacists, so not really like kind of and a lot of manufacturing, but a good chunk of it, about a quarter of it or a third of it, is in that bioanalysis drug development. What was a big change this year from last year is before they had combined what they called the Farm 360 NBC, or National Biotech Conference, and that was a focus on large molecules. So they kind of, sometimes people overthink things. So they had functions separately for like seven, or the NBC conference, the AAPS conference been going on for a long time. I don't know how long, but it's
Yeah.
easily
Yeah. Yeah. Yep.
20
Yep.
or 30 years. But the NBC piece was maybe less than a decade old, and they wanted them to come together because I think people said, "Oh, I don't want to go to two." Well, that was a disaster. For about 3 years, they were together, but it turned a lot of the biologics people off because they were overwhelmed with pharmacists and CDMOs, and now they've switched them back out. So I think that's a good move. But we
Mm-hmm.
still are always going to go to AAPS because there's a lot of focus groups, and I missed it this year, but I get to meet I'm on the PK focus group, I'm on the NAB assay focus group, and I'm on the liquid matrix, non-liquid matrix biopsy groups. Well, those groups met there, and that's unfortunate, but that's another opportunity to meet people that you collaborate with and you publish with, and it's always a fun time to go grab lunch or dinner with those groups. So that's something I missed, but I think that's an important part of AAPS or Farm 360. Is there's so many communities and focus groups that we participate in that we can go and just meet them in person and talk about the future. So that is something that Farm 360 brings a lot of value to.
Sure. Yeah. Absolutely.
And
I, yeah, Farm 360 really, it's a interesting blend of a big exhibition, and then then like you say, there's the sessions that are sort of like satellites around the exhibition hall. So
you know,
it's a valuable meeting. It's a really good meeting to catch up with people.
historically it's been
I
the most glitzy of, like, historically it's been the most glitzy one, when I say, or glamorous
Yeah.
one, right? Sometimes there's often a lot of, like, one year I got to hold an alligator, or every group tends to have some sort of nice prop or tchotchke or coffee, right? Or that was the one
Yes.
thing I remember most. It was probably our most glamorous meeting, where everybody rolled out the red carpet. I don't know. COVID has changed everything, I think. I don't know what it was like this year, but anyhow, John, maybe we can move on from Farm 360.
So yeah, so we I mean, like we participated in a couple of immunogenicity conferences, which obviously I'm not and
Yeah.
we're part of that team. So I'll pass that back to you.
The Immunogenicity and Bioassay Summit, actually, Dr. Sheck Kane from KCS was heavily involved there. He was on a panel, gave a couple talks. He actually worked with Stephanie Passes Pharma, who we've previously had on our podcast here. She's a member of our scientific advisory board and a consultant in the immunogenicity. She's a consultant in a couple different areas, but really emerging as somebody who's helping with immunogenicity and gene therapies and stuff. They gave and really gave they presented on what I had touched on in referring to WRIB. It's this really strategic changes in preclinical immunogenicity. So if you're giving in the past, if you were doing a GLP tox study, you had to invest copious amounts of time and money into a 3-tiered, right?
Yeah.
Immunogenicity methods have that screen, confirm, titer. You had to do full validations of all 3 tiers for mouse and non-human primate or whatever 2 species you were going into. And now they're backing that off. Now, even in some, like if you have a monoclonal antibody that's known to be non-immunogenic, or you've got approved versions of whatever you're manufacturing, you can actually get away with just having a developed method or a characterized method or qualified method and a single tier, and then just one second tier. And they use signal to noise ratios, and that you don't ever have to test a sample. Some people are doing it that way, John. They're just saying, I've got the method ready. If my PK analysis is perturbed, I'm going to go ahead and do it on my ADA method to confirm it is indeed an ADA and not something else. Because remember, you don't even need neutralizing. That's not what I'm saying. But they use a combination of your PK stats along with just having the method ready and then not testing samples. Well, that's a very big financial benefit and doesn't really affect the quality of the drug. More importantly, it's not just the money of it.
Sure, absolutely.
That's only part of it. It doesn't bring any benefit. But that's one strategic. And then, of course, if you have an ADC or you're getting into like any sort of bispecifics now and things that have they fall into like a mid, I call them a middle or moderate immunogenicity. Well, that you don't necessarily have to fully validate everything, but that is more, you can qualify the method, you get 2 tiers and you test the samples. And then there's high risk. And high risk is like enzyme replacements and things like that. You should invest in a validated method because typically you're treating rare diseases. So you're going to use your preclinical data as a big part of your the life cycle of the drug versus, your clinical trials your phase 3 trial might have 10 people in it. So you're going to have to invest more heavily. So that's the logical progression. But what I really want to hammer home is this is great news. But with that, the FDA in return has said, when in the past you didn't have to fully validate your ADA methods until you got into phase 3. Well, now they're saying out of the gate, your first in human studies, you need a validated 3-tier ADA method. So it's a give and take.
Oh,
right?
wow, okay.
Yeah, it's a give and take that happened here. It is and I want to emphasize the FDA. The FDA has been, to me, the FDA in the last 5 years has become much more logical. Not that they were illogical. I hope that didn't come out wrong, but they're really taking a proactive approach to try and cut down the expenses of what they're asking people to do because they're cognizant. And it's, again, I keep harping on money, but what's it really doing? It's delaying getting valuable life-saving drugs to the market because of cost. So they're linked slightly, right? But that is a big change.
Yeah,
clinical methods do not need to be done. Go ahead. Yeah, go ahead.
Yeah, you certainly don't want to be wasting your time on studies that aren't going to teach you anything. So yeah, absolutely, they want them to be thinking nimbly on this kind of stuff, which is why things like WRIB and EBF are important, because it's the industry will also discuss this. Plus, there's usually especially at WRIB that ability, the opportunity to interact with the regulators. So this stuff has been discussed,
That's a great
time issue.
You make a great point, John. It was about 3 or 4 years ago that people started opining about why are we doing validated GLP ADA methods for GLP tox studies. They're not needed. And here we are 3 years later, the FDA heard them, and it started with some harmonization papers and recommendations that the FDA takes very seriously. So that feedback changed the field. So those of you that are in the field listening, raise your hand, make your voice heard, and you can get what you need. Like I said, there's much more flexibility with the FDA. But John, we got one more immunogenicity conference, and then we'll move on to we'll give you the last stage with EBF. But
you let me speak there. Thank you.
So the immunogenicity and bioassay summit, that one I think is like the 8th or 9th year. I've been to about maybe 6 of those. But this is the 3rd annual Gene Therapy and Immunogenicity Summit. So this was new to me. And John Pirro, who was on our show last week, he attended this one, and I heard nothing. It was really only a couple hundred people, but it was a who's who of immunogenicity and a hard-hitting conference.
Mm-hmm.
And what I mean by that is it was all like, really people that are responsible. And it's not just a title. They're the people that are responsible for their organizations, and this is gene therapy companies specifically. A lot of it, when I say gene therapy, it gets lumped into CAR T-cell companies because technically it's a cellular gene therapy combination. But this meeting, I mean, people are there really. There's a lot of people who are in the cell and gene therapy space and don't really truly understand the immunogenicity component of it. I think they understand some of it, but it's really an evolving thing where you have a humoral response and a cellular response. And I think the people who are heavily into cell and gene therapy, they understand the cellular. Am I augmenting the patient's T cells to attack other cells, or are the cells I'm putting in there attacking other cells or healthy cells? Likewise, gene therapy companies understand, are they augmenting to have cellular responses? And so it's that anti-drug component that gets them a little bit and that's the humoral piece, and that's the antibodies that are produced. And I want to be careful not to say anti-drug, because that's not, anti-drug would be an antibody against the cell or gene. That's not really what happens. It's usually against what they're trying to knock in. So the transgene, as well as the viral vector or whatever vector they're using, not necessarily the actual drug, because a lot of gene therapies are packaged in nanoparticles and things like that. So it's a really complex thing, and I'm going to try to summarize what I'm saying. But from what I heard, it was a lot of learning, a lot of people, a tremendous amount of discussions around you looking at anti-therapeutic antibodies against the vector, which is often a viral vector or an AAV,
Sure.
right? An adeno-associated virus, and then that transgene. So if I'm given an AAV with a piece of genomic material to transcribe, I'll just say insulin or whatever sort of protein I'm looking for, now you got to worry about anti-therapeutic antibodies against the transgene or the protein you're knocking in. So great stuff. It's really a fascinating, you can tell I'm a little bit into it. John, I had a dream. I dream about it sometimes. That's how much this is on my mind.
So let's move on to.
That's so sad. I literally sometimes wake up.
Move on to EBF.
Sorry.
We'll try and calm you down, and we'll move on to EBF.
Yeah, please do.
So this is, I think of all the lists, this is the one it's still to come. It's actually happening mid-November in Barcelona. EBF is the European Bioanalysis Forum, but this is actually their open symposium. European Bioanalysis Forum is actually an industry initiative where it's actually companies in Europe, be pharmaceutical companies and CROs get together to discuss topics, and then the open symposium in November is really more of a meeting where other people so you can only be a member of the European Bioanalysis Forum if you're based in Europe. So KCAS, as it stands, couldn't be a member of the European Bioanalysis Forum, but we can definitely attend the open symposium because we then have that opportunity to discuss with European participants, etc. So it's similar to WRIB in terms of it's a very focused meeting. There is a small trade exhibition, and they have it planned such that when the talks are on, the exhibition hall is empty. When the lunches and other social events tend to be in the exhibition hall, and there's no talks going on at that time. this is a bit. I like I mentioned before. I've been loads of times to EBF. I think I've missed maybe two of them, and this is. I'm not sure what number we're up to now. Maybe the 15th. Um, this is a but however this is a big year for KCAS because we had a couple of I and Jeff Goddard attended last year, and that was the first year in person after break due to COVID. This year, with a recent acquisition of active biomarkers, we are sponsors for the meeting, so we're actually going to have a joint stand with active biomarkers and KCS, so people can come and find us and talk to us about the business. So this is a big step forward, as opposed to we were just general attendees last year. This year we actually have a presence, and then the other piece of that is I'm currently about 90% through it, but I will be doing an oral presentation on the Thursday of the conference, talking about some of the work that's been done by Habibi Gudarzi's group, and hopefully touching on some of Don's group stuff, just as a final footnote to the talk. That if Don has the date, it will make a really nice story. If we don't have it, then it will just be the Habibi's work on complex peptides and oligonucleotides and things like that. So I'm actually really looking forward to the talk. One of the things that EBF was always good about, and then you say about how things changed due to COVID. It used Pre-COVID, they would always have cocktail hours at the end of the day, which were really nice sessions, a good chance to catch up with people. Last year, the cocktail session was knocked on the head. This year, they're still thinking about do they bring it back or not. So fingers crossed they do, because it was always a nice way to get the evening started, and then you jump in a taxi and go and find a nice restaurant in
Yeah.
Barcelona for free. So
I don't
that
know
was
how you don't have that. That's hard. That's work hard, play hard. So I hope you get to do
yes,
that.
yes, exactly. But it's a really good meeting. Lots of I really. Oh, and I want to talk on the opinion stuff, in terms of thought leadership, like we touched on earlier with WRIB and that ability to talk to the regulators. EBF was very big on tiered approach to biomarkers, sort of not doing full validations for everything, really doing fit for purpose, whether you qualify it, validate, partial, do
Mhm.
abbreviated validation. And then another nice initiative within EBF, they have a young scientist session. So the young scientists will have other meetings during the year, but they actually have one session in the program will be young scientists presenting their work. And certainly last year, there was a real focus on how do we make bioanalytical more sustainable. So playing into what we were talking about as the first topic, and bio compostable masks and biodegradable gloves, EBF is actively talking about how can we make bioanalysis a more environmentally friendly initiative. What the hell? What's the word?
A greener, right?
More friendly. So that's. But so it'll be interesting to see what other things that will be discussed this year. But it's a very good meeting.
Yeah. No, I'm excited for our second year attending, right? And excited for
Yep.
you personally. And really, you said something that should be emphasized. It's a we're co-presenting what our active or co-exhibiting. That's the word.
Hosting,
Co-exhibiting
yes, yes.
co-exhibiting with our active biomarker um, brethren, and so they've been a lovely addition. Really excited, been working with them, and I shouldn't forget our Flowmetric team. Actually, it is. I did a conference. Part of the reason why I missed some of the other conferences is I did a Philadelphia Life Sciences or Pennsylvania Life Sciences event as well in October. So I gave a small little, not a small, a a session on discovery that we spoke on. Myself, Ren, the founder of Flowmetric, and one other individual external to us. So like I said, this is a very active time of year as we kind of, I hope, hashed home to everybody. But John, I think we're wrapping up, and you know, I see that we have a very funny. We got some feedback. We haven't done feedback in a little while, but John, do you want me to read this feedback, or do you want to?
Yeah, I'll just give a final thought on
Okay, sure.
EBF. I'm very much. It will be a good meeting, but it's for us. It will be excellent to be co-exhibiting with active biomarkers, because it's one more step towards integrating both companies and actually building that teamwork across the Atlantic. And having the opportunity to share booth space with them will be great, and certainly it will give us a real leg up in terms of boosting KCAS's profile in Europe, because as a company, because we've really been US focused, we haven't spent too much time on Europe. This will change things markedly for us. So I think it will be great.
Yeah,
So yes.
yeah, absolutely. And with that, John, as always, a very nice session there. But we're in order to maybe not run too much longer. I see we're not doing too bad on time, but this is a funny one. I'll read it. Riley says, "Hi guys, this is the first time I am contacting you. So first off, I'm a huge fan. Love the podcast. My only suggestion is record more often. Ha ha ha. So, and then Riley continues and says that the reason for the note is I want to be delicate in the way I word this, as I am in no way disparaging Dr. Pirro, although John he did a fine job. But you can't just swap out hosts on us without any warning. Please say that Dominic will be involved in future episodes." Well, I think I hope we've answered that. The answer is yes. I'm flattered. Thank you, Riley, for the kind note. And I'm not going anywhere. I do enjoy doing this. I just happened to miss one of our windows because of the conference and traveling and so forth. So that was why I wasn't able to do last month's podcast or last podcast. And so here I am back for the 61st episode. And as far as I know, I'm here for the foreseeable future, but maybe John Perkins would like a different co-host, one that doesn't harp as much as I do about immunogenicity and Lord knows what else. But thank you again,
Well,
Riley, for those kind words.
See, I love it because I learn stuff on this podcast every time, so it's all really, really valuable to me. So I'm definitely happy to have you co-hosting. Riley, it's just one of those things that sometimes it's always tough to find a slot. If you look at our calendars, you'd realize how many things we're juggling on a daily basis. So then to find a hour and a half slot to do this can be challenging, and then there's travel involved as well. So it's actually good to have John Pirro as a third potential presenter, such that we can slot in and out. We just don't. We've got no real knowledge of when or if it's going to happen. But sometimes it's you really have no choice but to do that.
Yeah, no, and I think we're recording on a Monday, which I think is our first. And not only is it Monday, it's actually Halloween, which is maybe John, instead of asking what you're going to be doing this weekend, do you have any Halloween plans?
No, I'm not really a Halloweeny type of
Oh, that
person.
surprises me, John. What do you mean that you don't have like at least 10 or 12 costumes that you can wear like I do? That's just shocking to me that you don't
No,
really.
that's what my wife does. She was out on Friday night as Ursula the witch from The Little Mermaid,
Oh,
and then I'm not sure. I've seen some makeup this afternoon, but I'm not sure what she's doing this evening. So, but I think she's the one that obviously loves opening the door and greeting people and all that. I'm very much more. Yeah, sorry, I whatever.
well, I love it. I get all into it, as does my wife. We had we go to a nice Halloween party. This was the fourth year that we'd gone to this one. Last year, I won a prize for best costume. I went as a ghost, and my wife was the Ghostbuster. And so it was if you had saw how I looked, maybe I showed you a picture of it. I was really
I think I've seen it.
I
Yeah.
was very much in a lot of makeup and almost looked like Uncle Fester slightly, except I had like a bunch of blood on me. But you can imagine what my Uncle Fester, if you don't know who that is from the Addams Family, is such a handsome guy. No, I digress. This year we went as I went as Mike Myers, and she went as the cheerleader I killed. So she got a little kind of made it look like she was kind of dead, and I just had a Mike Myers mask and a sword. And so I'll probably hide out. I typically like to scare the older kids a little bit. It's always funny when, if you're walking up to trick or treat and someone comes jumping out at you, it will scare you. I even ran last night. My son had a pumpkin carving party next door, and I went over and let them know that Mike Myers was still alive. So actually, it was Jason. Sorry, I keep naming the wrong thing. It was Jason from Friday the 13th. Jason Voorhees there.
Oh, okay.
Yeah,
Okay.
not the wrong one, but we.
Yeah. So
Go ahead.
go on.
No, I just. I love Halloween.
Tonight I'll be
Go ahead.
So tonight I'll be relatively quiet, but tomorrow I jump in my car and drive to Boston to see the Chameleons play Brighton Music Hall, and then I drive back on Wednesday. So that's what I have to look forward to.
Yeah, that's your favorite.
And then the other bit of news for us was my daughter's finally named a wedding day. So September 2nd next year, we'll be having a big family day. So now we start into planning mode. So my wife is already spending all hours online trying to figure out stuff for next year.
That's awesome, John. Congratulations. Your family's growing and continues to grow, and that's very exciting. For me, that big green egg I got a couple weeks ago or a couple months ago now is something I dream about that as much as I do about immunogenicity of gene therapies. But I've got some neat little. I got this brisket I got my eye on for the upcoming weekend. It's one I got. I've been shopping a little bit more locally, going to more farmers markets, just kind of, I don't know, it's a cool way to shop. And so I picked up this awesome brisket of, I think it's a big one. It's probably, I think it's like 5 pounds, good size. And I think I'm going to start smoking it maybe at like 5 a.m. and probably do a good at least 12, 15 hours or something. We'll see. But I'm excited to do that. And just the green egg in general has been so much fun. And then I made a batch of sauce. You can't go without talking about pasta and pizza. And so it is a time of the year where I made a batch of pasta sauce back in August and canned some of that. And got my eye on maybe making another one. But it's been some of my the weekends have been recovering from traveling, John, right? So less food on the mind, but
sure.
Getting back into it this time. So we're coming to an end, but why don't you want to tell? We should definitely mention that.
So I'm just going to quickly say, so the weekends piece, for us, the last 3 weeks, we've actually had my wife's biological mother and brother staying. So they flew back on Tuesday of last week, and in true airline form, managed to get delayed leaving Ithaca, so they missed their flight out of Newark, and finally got back 24 hours later than they should have done. But then we hosted them for 3 weeks, which was really nice. We're going into a little bit of a quiet phase before then I travel over to Europe. Obviously for EBF, but I'm also doing some family stuff around that. So it's up and down in terms of things.
I think the reason why I missed the last podcast was I was in Vegas for celebrating my 50th. So that was another thing that I'm not going to tell you too much about that on the air, but it was a lot of fun, and I didn't do too much out of the norm other than it's Vegas, and it was a lot of fun. I believe that was why I missed the last podcast.
Yep.
And so it's good to I would like to get to some normalcy, and certainly I think we'll have Thanksgiving before the next podcast. Speaking of the next podcast, we do have a topic for the next podcast. The 62nd one this is an exciting one, right, John? We're going to feature Mark, who is the general manager of active biomarkers, and John Bucksath, our CEO of KCAS. So this is going to be an introduction to active biomarkers. If you're a podcast listener, that will be an exciting one. The 62nd one, as I mentioned, will feature the general manager of Active Biomarkers and the CEO of KCAS. Should be a good one, John. Very excited for that.
Exactly. Yes. And I'll have lots of news from Europe by the time I get back.
Yeah,
I'm sure.
With that, we'll see you in the funny pages, I suppose. Take care.
Yeah, absolutely. Yep. Thanks very much.